Good morning. I'm Joe Alvarez. I'm an associate clinical professor at the City of Hope, a comprehensive cancer center in Duarte, California. I'd like to welcome all of you to this third press briefing of the 60th ASH annual meeting and exposition. This one focused upon CAR-T cell therapies, new research and updates from pivotal trials identified from the submissions for this meeting. Before we begin, I'd like to ask you to please silence your phones, and if it's possible to silence the shutter sound on your camera. I'd also very much appreciate it if you could do that. The studies presented today spotlight the continued success and the realization of the promise of this technology, chimeric antigen receptor T cells, or CAR-T cells. And as you may see reflected throughout this meeting, immunotherapy for the treatment of blood cancers has been identified as one of ASH's 6 research priority areas. And this is reflected in the ASH agenda for hematology research. As you've likely heard, this agenda serves as a roadmap for the best path forward in helping to prioritize research really directed at helping to navigate funding agencies and foundations that can in turn equip researchers today and in the future to make practice-changing discoveries like those that you're hearing about today. Now, parts of what's unique about this therapy is that these therapeutics, in fact, are designed by harvesting a patient's own cells and re-engineering them, re-engineering and manipulating the immune system to target specific proteins found on the surface of leukemia or lymphoma cells, respectively, and then subsequently reintroducing these T cells back into the patient's own body, where they can supplement the activity of the immune system and target these tumor cells that may have proven refractory to multiple forms of therapy prior to treatment. So today we'll be hearing about the success of these novel therapies with extended trial data. We'll also hear about work intended to augment CAR-T cells with the administration of additional oral drugs or immunotherapies. Before introducing the respective speakers, I'd like to congratulate not only the speakers but the groups that they lead and represent in the extraordinary work that they've done to help bring these new therapeutics to patients and to better understand the nature and potential of these therapies. Dr. Stephen Grupp at the Children's Hospital Philadelphia will present the results of long-term follow-up of the Eliana trial. This is an international phase 2 study that provides data on an additional 4 patients, as well as 2 additional years of follow-up data that shows among 79 pediatric patients with relapsed and treatment-resistant acute lymphocytic leukemia, over 80% achieved complete remissions after receiving a single infusion of a CAR-T cell therapeutic, tisagenlecleucel, which targets a CD19 protein frequently found on the surface of these cancerous cells. A year and a half later, 66% of patients were still in remission, and the median survival in this trial has yet to be reached. Details on this can be found in abstract number 895. Dr. Thomas Maziarz from Oregon Health Science and Knight Cancer Institute in Portland, Oregon, will present the long-term follow-up data from the Juliet trial. This is a global pivotal phase 2 trial of CTL019 in adult patients with relapsed and refractory diffuse large B-cell lymphoma. 19 months after receiving a single dose of tisagenlecleucel, again, a CAR-T cell therapy that targets CD19 proteins frequently expressed on cancerous lymphoma cells, a high rate of response persisted over a year and a half amongst adults with relapsed and refractory diffuse large B-cell lymphoma. The data from this abstract will be presented in poster number 1684. Dr. Jordan Gauthier from the Fred Hutchinson Cancer Research Center in Seattle will present a phase 1-2 study investigating whether continuing to administer the oral drug ibrutinib before, during, and at least for 3 months after CAR-T cell therapy improves outcomes in relapsed and refractory and difficult to treat chronic lymphocytic leukemia, or CLL. Dr. Gauthier will be reporting on an improved response rates and diminished adverse events in the cohort that continue to receive concurrent ibrutinib. Details on this presentation can be found in presentation number 299. Dr. Shannon Maud from Children's Hospital Philadelphia will present a first-of-its-kind study on the application of a PD-1 or checkpoint inhibitor to heavily pre-treated pediatric patients receiving CAR-T cell therapies for relapsed B-cell acute lymphocytic leukemia or lymphocytic lymphoma. And her finding is that this can extend the treatment response. More than a year after receiving checkpoint blockade inhibitors, half of patients exhibited either partial or full responses to therapy. Details from this abstract can be found in abstract number 556. And finally, Dr. Corinne Summers of Seattle Children's Hospital and the Fred Hutchinson Cancer Research Center will present results from a phase 1-2 study investigating the role of hematopoietic stem cell transplantation after CAR-T cell therapy on sustained remissions. Across several cohorts, there was a trend towards improved disease-free survival in patients who underwent stem cell therapy following CAR-T cell infusions. Dr. Summers will have more to say on how each cohort fared, and details can be found in abstract number 967. So as you're seeing, we're living in an extraordinary period of innovation, particularly in this domain of CAR-T cell therapeutics. Yet there are some challenges ahead, inasmuch as we're trying to establish and understand the best paths forward in using these therapeutics. We've also run across the more prosaic and grounded issues of how do these T-cell therapeutics get paid for. There are challenges with respect to reimbursement, and we've seen a policy, the Inpatient Prospective Payment System rule that CMS produced earlier this year, that creates challenges to centers that are delivering these therapeutics due to a lack of suitable and sustainable reimbursement for the delivery of these therapeutics. Now this is an important issue, and I don't want to sidetrack the greater topics that we're talking about here, but even Dr. Scott Gottlieb, FDA Commissioner, has noted in public on 2 occasions that unless we're able to navigate a reimbursement system, that it does pose a challenge to the level of innovation and the sustainable pace of innovation around these therapeutics. ASH and other organizations are working in collaboration, meeting with CMS and legislators, trying to address this crucial issue while our researchers continue to advance the state of the art. After all the presentations, we'll open up the floor to questions. To ensure that we have plenty of time for all the presenters, we're going to ask you to hold your questions until everyone is done, and then we'll navigate your questions. I'd like to start with Dr. Stephen Grupp with the first of the presentations, and we look forward to entertaining your questions.
Okay, thanks very much. It's a real pleasure to talk about our work, and also to see a session focused entirely on CAR-T cells is really exciting to me, and represents, I think, the value that ASH puts on these new clinical approaches. And so this is absolutely an update to a registration trial for what is painfully named tisagen-lecklucel, previously called CTL-019, and FDA approved as Kymriah. And I'm speaking on the behalf of an amazing group of international investigators who participated in this trial in 25 centers in 11 countries across the world. And so this is was the first CAR-T cell trial with a potential registration endpoint, the first global trial, the first trial in which there was a global supply chain from the drug company that was commercializing the product, which was Novartis. And this was in patients under 25 years of age, and these patients essentially formed the basis for the FDA approval in August 2017. After this, we'll hear about the further approval in lymphoma, and this was the same product, just to be clear. These 2 talks are about the same product. And now this has moved forward to approvals in the EU, UK, Canada, Switzerland, and we'll see about other countries as we try to approve global access. These were relapsed and refractory patients. Eligibility criteria, ages 3 to 21, although that was 21 at diagnosis, so patients could be a little bit older. They had to have morphologic disease. We didn't allow isolated extramedullary disease or prior anti-CD19 directed therapy, which included the drug blinatumomab. The overall endpoint was remission rate within 3 months, either CR or CRI with incomplete count recovery. And this update is really 11 months further follow-up in this group of patients. So I think that this and 2 slides from now are the key take-homes from what we're presenting here. And so these curves, the first curve is the duration of remission curve. And I want to be clear about the fact that we're taking patients who have responded to the therapy and ask the question, how are they doing over time? And now with more follow-up, what we're seeing is patients who go into remission, which is 82% of the patients, remaining in remission at 12 and 18 months about 2/3 of the time, 66%. So that that longer term follow-up, I think, is very exciting to us and indicates that there are a group of patients for whom this therapy, most of these patients did not go on to transplant, and we'll hear more about this in the future. This is a therapy that has the potential for long-term disease control. And then if you look at all the infused patients, the other curve is the overall survival curve, and again, we're seeing excellent overall survival in these patients. And so we're pleased by this long-term follow-up data. The overall survival numbers at a year, 76%, and at 18 months, 70%. So quite consistent with what we're seeing in the duration response curves as well. Obviously, these are two slightly different analytic populations. We're really not seeing anything new in terms of toxicities. We see the significant toxicity of cytokine release syndrome. Up to 50% of patients have significant cytokine release syndrome that was controlled by the toxicity management protocol that was developed for this protocol and has been used in a number of treatment protocols and commercial settings since then. Low infection rate. There are some patients who have cytopenias that don't resolve, so grade 4 cytopenias in 18% of patients at day 28. Very low neurologic events, no grade 4 neurologic events, no cerebral edema. So this is really, this has been consistent throughout the CHOP studies, the PEN studies, and the various Novartis studies in terms of the toxicity. This is the other, I think, fascinating new piece of information. The 82% of the patients who went into remission were all but one were negative for the typical approach to MRD, minimal residual disease testing that we do, which is multi-parameter flow in the United States. This is another assay, a next generation sequencing assay that is more sensitive, at least 10-fold more sensitive. And here we're asking the question, what's the impact of being sensitive by this next generation sequencing assay on these patients who are in remission? And so you see this really significant curve separation between patients who are positive and negative. So patients who are negative by this assay, more than 80%, about 80% of the time remain in remission. And if they are positive, more like 20% of the time remaining in remission. This is quite striking, and this is also holds up in the overall survival analysis. And so this is brand new data. And that green curve, as we think about the role of transplant, and again, you're going to hear more about this, this is a group of patients that I'm wondering whether they actually need transplant, and the vast majority of them did not get one. So, clinical outcomes. The other sense that we've gotten is that patients with high degrees of CRS also have higher degrees of neurologic events. So we see more grade 3 neurologic events in patients who have grade 3 and 4 cytokine release syndrome. Again, we didn't see any grade 4 neurologic events. The relapses as a transplanter look like relapses after transplant. So if you're not relapsing by a year or two, the likelihood of that happening is a lot lower. So it happens within the first year. Most of these relapses are immunological escape, CD19 negative, and those patients who have CD19 negative ALL still have CAR-T cells that are still working for the patients, but they don't prevent relapse because the ALL is actually preventing, it has lost the CD19. And then I already mentioned the MRD analysis. So we think the efficacy is sustained and excellent to 82% of the patients achieving remission. These are durable CRs, even without further therapy, haven't reached median duration of response or overall survival. We can manage toxicity, and I think this is clearly an opportunity for us to treat our patients both in America and now across the world. Thank you very much.
To present next.
Yeah, I want to thank you all for being here, and I have the pleasure of representing our group, our investigators from multiple countries embarked on the study of tisagenlecleucel in large cell lymphoma, and this is a longer-term follow-up of the preliminary early data that was presented last year at ASH. Just to state that this is, again, you saw some of these, the early part, tisagenlecleucel is the first approved gene therapy, the first approved CAR therapy in the United States. May 1st of 2018, it was approved for a large cell lymphoma. It is a single-arm, open label trial, so there's no randomization. We know where patients are getting, and we will update this analysis as this is 19-month median for follow-up. The reason this is important is that early on, when you look at data early, you can always worry that the trial has selection bias. You pick the best patient as it doesn't represent real life. As you move into 19 months, follow-up, the median 19 months, you are now looking at the real natural history of disease. And these are patients with multiply relapsed refractory large cell lymphoma. The primary endpoint was best overall response, which is CR and complete remission and partial remission, with key secondary endpoints of duration of response, overall survival, and safety. Now, just for the event, this will be presented tonight in poster session. I'll be there for most of it, or Dr. Stephen Schuster, who's our first author in the publication. This is just to announce that at 9 o'clock this morning, this paper has just been published by New England, it was announced for publication by the New England Journal of Medicine. As far as the data, the overall response is 54%, of which 40% were complete remissions, 14% were partials. Of the patients who had partial remission, we do know that half of them went on to develop complete remission over time. So the drug continues to work. Again, over time, you can convert from partial to complete. The overall response data was seen. There's multiple subgroups of large cell lymphoma. It's a very complex disease, but whether all the potential prognostic subgroups, we could not see a predicted group that was likely to be favored or also predicted to fail with CAR-T. Also important is the median duration of response was not reached for the patients who were responders, and for patients who were in complete remission, over half of them, the median remained free and alive, with the median overall survival not being reached. And for all patients infused, it was 11.1 months. The figure on the right actually compares as an overlay of the data from the Juliet study compared to one prospective as well as one retrospective study of similar patient population. And again, early on you have patients that will fail, but what we see is we're approaching, again, a plateau, but we're definitely seeing a change in the natural history of the disease to the best of our ability to interpret the data. As far as safety, we have similar profile, but not to the same degree as tisagenlecleucel when treated with acute lymphoblastic leukemia. It's a lower degree of the cytokine release syndrome and neurotoxicity. And this may be in effect relate to the fact that when you infuse the cells, they don't immediately encounter in the same vascular space their target antigen. They have to traffic out of the vascular space into actually a lymph node or spleen or other organs. And so this may, is hypothesized, may at least contribute to lower degrees of the adverse events seen, but we still do see 25% with advanced grade 3, 4 GVHD. And again, I would also share that these grading system is a very liberal system that is that over-identifies toxicity compared to the Lee criteria, which has now become more generally utilized. Since the previous report, no new deaths of a cause other than due to any cause other than one patient with disease progression. No treatment-related mortality was seen throughout the entire study. And there were 3 early deaths, all related to lymphoma that progressed through. So in conclusions to share with you, this is the data with a longer median follow-up of 19 months. Just to say again the high points, the overall response was 54%, and for patients responding, the median duration response was not reached. In a similar population of patients, we would have predicted a less than 10% complete remission and a 5-month median survival. So thus it was improved over historical options, and I would say the adverse events were similar. And this is just a final slide to say that when you begin work, I mean, this is the beginning, not the end. You know, what we are doing is hoping you'll hear about ways that we can build on primary first-generation T-cell therapies. We have 6 current projects that are coming out of this database. Some looking at dosing rationale. We are doing 2 presentations on Monday. We'll be looking at regrading toxicity using the Lee scale or the neurotox scales that have become more commonly used. We're also doing work on health economics as well as examining what happens when the cells are given in remission. So I want to thank you for the time and appreciate the effort, and I also will vouch for Dr. Oliver. And this is the importance of this has become a health economic issue that we all need to be aware of and highlight. Thank you.
Thank you very much. I'd like to ask Dr. Gauthier to come and deliver his presentation.
Okay. Good morning. I would like to thank the organizers for inviting us and to participate in this press meeting. So we've heard about CAR-T cells for ALL, CAR-T cells for diffuse large B-cell lymphoma, and now I will try to show you how we're trying to improve CAR-T cell therapy for a different kind of disease called chronic lymphocytic leukemia. So the key point is that in most cases, this is a slow-growing hematologic cancer involving B cells, so they express the CD19 antigen, and it's actually the most common leukemia in adults, and the common symptoms are elevated lymphocyte counts and swollen lymph nodes. And the problem that rises is when patients do not respond or relapse after conventional therapies, and prognosis can be poor, in particular in patients who do not respond or relapse after an oral agent called ibrutinib, which prevents the CLL cells from growing by inhibiting an enzyme called BTK. What we've done is we've carried out a phase 1/2 study at our institution using a CAR-T cell product of a defined composition of CD4 T cells and CD8 T cells that is called JCAR14. We have published that this approach could lead to durable responses in patients with CLL that had relapsed or were refractory to ibrutinib. What could be the rationale to try to combine ibrutinib with CAR-T cells? There's actually a variety of reasons why you would want to do so. Because sometimes when you interrupt ibrutinib, patients may experience a rapid tumor progression that we call a tumor flare. We think as well that CLL cells ibrutinib can help mobilizing the CLL cells into the blood, and based on some preclinical and clinical data, there is evidence that ibrutinib may improve the function of CAR-T cells and may decrease the toxicity related to CAR-T cell therapy. I'm going to show you a retrospective analysis of 2 cohorts sequentially treated on this trial, including 43 relapsed refractory CLL patients with detectable disease after ibrutinib therapy. So the key point, in the no ibrutinib group, all patients had interrupted ibrutinib at some point before leukapheresis or before lymphodepletion. In contrast, in the concurrent ibrutinib cohort, all patients were scheduled to receive ibrutinib at least 2 weeks prior to leukapheresis, up to at least 3 months after CAR-T cells infusion. And the key distinctive feature between the 2 cohorts is that all these patients hence received ibrutinib from the time of lymphodepletion onwards. Of course, this was not a randomized head-to-head comparison, but most patients and disease characteristics were comparable between the 2 groups, and I will refer you to the full-length abstract. First finding is that the use of concurrent ibrutinib with CAR-T cells was well tolerated in most patients, and 13 out of 19 patients could receive ibrutinib as planned without discontinuation. Now, if we look at the response data early on at 4 weeks, the first finding is that this approach was highly efficacious, with 83% of patients achieving a response in the concurrent ibrutinib group compared to 65% in the no ibrutinib group. This is using a classification called IWCLL, which basically means that we observed a significant decrease in the percentage of CLL cells in the blood and in the size of the lymph node. And then further on, when we look using very sensitive methods such as flow cytometry and further deep sequencing, which can detect 1 tumor cells within 1 million cells, we observed that 72% of patients went into this deep remission by flow cytometry, and within these patients, 85% had no detectable disease in the bone marrow using this deep sequencing method. We know that CAR-T cells are important, and the kinetics of CAR-T cells in vivo is important to induce a response. Here I'm showing you the kinetics of CAR-T cells for 2 different subsets, the CD8s, to simplify, which are the helper cells, and the CD4 cells, the CD8s are the killer cells, the CD4 cells, the helper cells, and this is the kinetics, the number of CAR-T cells in the blood from day 0 to day 30. And here, this is only looking at the peak of CAR-T cells in the blood. And as you can see, there was a trend towards better expansion of CD8s and a significantly higher, better expansion of CD4 CAR-T cells in the concurrent ibrutinib group. We also know that CAR-T cells can cause toxicity. And we did observe toxicity in both groups, but strikingly, no patient developed a severe form of cytokine release syndrome in the concurrent ibrutinib group. We did note 2 fatal events, one in the concurrent ibrutinib group from a presumed cardiac arrhythmia, and 1 patient in the no ibrutinib group from CAR-T cell-related toxicity. In summary, in this retrospective analysis of 43 CLL patients with refractory or relapsed disease treated in 2 sequential cohorts, the concurrent administration of ibrutinib with CD19 specific CAR-T cells was feasible in most patients, induced high response rates and deep responses early on at 4 weeks, was associated with higher in vivo expansion of CD4 CAR-T cells and lower rates of severe toxicity. Next step is to hopefully validate these findings in a prospective phase 1/2 study. Thank you.
Thank you very much. I'd like to ask Dr. Maud to come up for her presentation.
Thank you very much. It's a privilege to present this work on behalf of our team at Children's Hospital of Philadelphia and University of Pennsylvania. What I'm going to be talking to you today is an extension of the work that you saw Steve Grupp present. This is data from the initial phase 1 trial of CTL019 showing relapse-free survival in this population of children and young adults with relapsed and refractory ALL. And we can see that in responding patients, the majority of patients will remain disease-free at 12 months with a 60% relapse-free survival, indicating very good outcomes, which you saw replicated on the global study of tisagen-lecklucel. And we have seen that in these patients who maintain disease response, the vast majority will have persistence of their CD19 CAR-T cells. And we can measure that by seeing that their normal B cells, which also express CD19, will be cleared from the circulation by the effect of the CD19-directed CAR-T cells. But for a fraction of patients, shown here, we can see loss of that B-cell aplasia. So when we see recovery of B cells, that is a marker of loss of persistence of the CD19-directed CAR-T cells. And we have observed a higher rate of CD19-positive relapse in that group of patients who lose their CD19 CAR-T cells prior to 6 months after infusion. So in this group of patients who recovered their B cells, indicating loss of CAR-T cells, we started to think about how could we potentially improve upon the persistence and surveillance for leukemia and hopefully prevent relapse. Our hypothesis was that T cells, upon activation, may become exhausted through activation of immune checkpoint pathways, and that one such pathway, PD-1, may be involved in early loss of CD19 CAR-T cells, and therefore that the combination with PD-1 checkpoint blockade may improve the function of the CAR-T cells and their persistence. So in the group of patients who had poor persistence or partial or no response to prior CAR-T cell therapy, we employed a combination approach with PD-1 blockade. With the schematic of the rationale being here, this is the tumor cell with the CD19 antigen and the CAR-T cell with a CD19 directed CAR. The T cell activation upon this antigen engagement results in cytotoxic effect and killing of the tumor cell. But what can potentially happen with T cell activation is that there is checkpoint activation as well, either through ligand on the tumor cell or on cells in the microenvironment, and that can then bind to the checkpoint receptor on T cells and block the effect of the T cells. So one potential way to block that blunting of activation is through monoclonal antibodies directed against the ligand, and another method is through monoclonal antibodies binding the PD-1 receptor on the T cell. And so this is the approach we took in 3 groups of patients where we hypothesized that this pathway may play a role. So the first setting was in patients who had partial or no response to CD19-directed CAR-T cells, where our hypothesis was that the activation of the T cells may lead to activation of the checkpoint pathway and block a full response. And in that group of patients, we added PD-1 blockade and saw that we unfortunately did not see an effect in this small group where 4 of the 4 patients treated had progression of their disease. One patient, this progression was marked by reduced CD19 expression, which was probably the mode of escape from CD19 CAR-T cells. But in the second setting, in patients who responded to CAR-T cells but had poor persistence, marked by early B-cell recovery, we saw that patients who were reinfused with a CAR-T cell product followed by infusion with PD-1 blockade, 3 out of 6 patients had a return of B-cell aplasia and sustained CR with B-cell aplasia, showing continued persistence of their CAR-T cells. And then in the third situation, this was a little bit different than the other 2. In this situation, we had patients who had bulk extramedullary disease, and we hypothesized that the PD-1 checkpoint pathway may be activated through the microenvironment in that extramedullary situation, and added PD-1 blockade for patients who did not respond in their extramedullary disease, and saw that 2 out of 4 patients had a sustained CR, and 2 out of 4 patients had a partial response with the addition of PD-1 blockade. We saw that this combination resulted in few adverse events, and it was well tolerated. A few patients did have fever or grade 2 CRS, as well as cytopenias, but overall it was tolerated very well without serious adverse events. So in summary, we show that PD-1 checkpoint inhibitors can be safely combined with CD19 CAR-T cell therapy, and that this mechanism may be useful to improve CAR-T cell persistence. And we found that in half the patients, PD-1 checkpoint blockade can improve that persistence, and that this patient may particularly benefit patients in that situation with poor persistence, marked by early B-cell recovery, as well as those with bulky extramedullary disease.
Thank you so much. I'd like to invite our final speaker, Dr. Summers, to come and deliver her presentation. Thank you again.
So I'd like to thank the committee for selecting our abstract, which focuses on our work evaluating the role of transplant for patients that achieve a sustained remission following CD19 CAR-T cell therapy in our Seattle Children's Plateau 2 trial. Our CD19 CAR-T cell trial is a phase 1/2 study in pediatric and young adults with B-cell leukemia. The study objectives include determining manufacturing feasibility of the CAR product, as well as determining the safety of the product infusion, as well as the product's toxicity profile. On the phase 2 portion of the trial, we're additionally evaluating the efficacy of the product. The study's eligibility criteria include age greater than 12 months and less than 27 years, subjects with relapsed or refractory CD19 positive leukemia, subjects with a history of bone marrow transplant could not have active graft versus host disease, and subjects also needed an absolute lymphocyte count greater than 100 cells per microliter. We treated 43 subjects on the phase 1 portion of the trial, which has been previously described. 1 million CAR-T cells per kilogram was determined to be the maximum tolerated dose, and this was the cell dose used for the phase 2 portion of the trial. The first 21 subjects treated on the phase 2 portion of the trial are described in abstract 4012. The 1-year leukemia-free survival for the subjects evaluated is 76%. However, we continue to see late relapses, including CD19 positive relapses at 22 and 27 months, as well as CD19 negative relapse at 37 and 41 months from CAR T-cell infusion. Therefore, we aim to evaluate the role of bone marrow transplant in maintaining leukemia-free survival and overall survival for subjects with sustained leukemic remission for greater than 63 days following CD19 CAR T-cell therapy. We retrospectively analyzed 50 subjects. 32 were treated on the phase 1 portion of the phase 1 dose-finding portion of the trial. The remaining 18 were treated on the initial phase 2 cohort. We excluded subjects who did not have an opportunity to undergo transplant. This included 4 subjects that did not respond to their CAR T cells, 9 that had an early relapse prior to day 63, and 1 subject that died from toxicity. We compared leukemia-free survival and overall survival between subjects with regards to the receipt of bone marrow transplant. When looking at leukemia-free survival for the subjects that did or did not receive a transplant appears to improve leukemia-free survival. We did not observe a difference in overall survival for the 2 groups, and we hypothesize this is due to continued responsiveness to therapy following post-CAR relapse, though more follow-up is needed. We then evaluated subjects without a history of transplant. It seems much of the transplant benefit observed on the previous graph stems from the population that received their first transplant. Here, the blue line represents subjects that received their first transplant following CAR therapy. The gray represents those that did not pursue a transplant. Of those subjects, 2 relapsed. Only 1 subject that received a transplant has relapsed. Though these numbers, there are a small number of subjects, the data is suggestive of a leukemia-free survival benefit for those without a history of transplant who pursue a transplant once in remission following CAR T-cell therapy. Again, we did not observe a difference in overall survival. Evaluating only the subjects with a history of transplant prior to CAR T-cell therapy, there does not appear to be a clear benefit to pursuing a transplant following CAR T-cells. The blue line above represents subjects that received a second transplant. Of the 10, 1 subject died from transplant complications and 4 subjects have relapsed. Of the 24 subjects that did not receive a transplant, 17 have relapsed. Overall survival between the 2 groups did not differ. Lastly, we evaluated only the subjects with short functional CD19 CAR T-cell persistence, demonstrated by return of CD19 positive cells by day 63 following cell infusion. Comparing subjects that did and did not receive a transplant, there appears to be a benefit to transplant for these subjects. As shown in gray, all subjects that did not receive a transplant ultimately experienced a relapse. Of the subjects that received a transplant, shown in blue, only 2 have relapsed. 1 subject died due to transplant complications following their second transplant. Again, here, overall survival was not statistically different. In conclusion, despite initial remissions, late leukemia relapse remains a barrier to long-term efficacy following CAR T-cell therapy for B-cell leukemia. Patients without a history of transplant who achieve a remission following CD19 CAR T-cell therapy may benefit from transplant. The role of transplant following CD19 CAR therapy for patients that have a history of transplant is unclear, and larger studies are needed. Transplant may abrogate the increased risk of relapse due to early loss of the CD19 CAR T-cell persistence. And overall survival differences were not observed, perhaps due to response to salvage therapy, though longer follow-up is needed. Thank you.
Thank you very much. We now have about 20 minutes for questions. I invite members of the audience. Yes, sir.
Neil Osterweil with Family Practice News for Dr. Mazarias. So you had 4% neurologic, grade 4 neurologic adverse events in the trial. Were there any cases of cerebral edema? I might have missed that.
The neurotoxicity, and we were actually regraded this with a group of experts, and I was saying, thanks we have a presentation on that on Monday, using the Lee criteria and also using the new CAR-Tox criteria. A lot of neurotoxicity identified as headache, lassitude, but the overall neurologic toxicity, there were patients with encephalopathy, but it was approximately, I believe, 12%.
Encephalopathy, but not edema.
Encephalopathy.
Okay, thank you.
Yes, please.
Hello, Linda Williams from Edinburgh News. I wanted to ask Dr. Maud, which PD-1 inhibitor did you use, and do you think there's a wider role for PD-1 inhibitors in this with CAR-T therapy?
We used in 13 of the 14 patients, they received pembrolizumab, and 1 patient received nivolumab. And what was your second question?
Do you think there's a wider role for PD-1 with CAR-T?
A wider role? So I think that we're seeing some encouraging results showing that there may be some patients who benefit from that combination, but larger studies and more data are needed to determine if there is a broader role for this.
Great.
Thank you. I have 2 questions. One is about transplant after the CAR-T cells. So Dr. Grupp, I think you said at the end of your talk that the responses are so good and so durable that it looks like they may not need a transplant. And then Dr. Summers, you suggest that maybe they do. So maybe you could both address that question. My other question, if I can ask Dr. Maziarz, is that right?
Maziarz, but
thank you. Can you comment on the other CAR-T cell?
Sure.
In the same, in lymphoma, in the same thing? And are the responses and side effects similar or not?
Yeah, so why don't I start out, since I think that's what you started with. So I think the role of transplant after CAR-T is one of the areas that produces the most passionate debate. I am actually a bone marrow transplanter by trade, and I actually find the idea of not being able to transplant a large number of these patients to be very attractive. But I think the performance of these cells differs very much. The clinical trial setting is also somewhat different. Comparing trials to trials is sometimes a challenge, but I think the thing that has emboldened us to avoid transplant in 80 to 90% of our patients is really the issue of persistence. And so you saw Shannon's B-cell aplasia curve, which applies to our data, and because so many of these patients keep their CAR-T cells, this is the group of patients we've really focused on. We agree very much that patients who lose their B-cell aplasia need more intervention before 6 months, so I think there's agreement on that. And I think all of us are like very uncertain about the issue of if you've already had a transplant, you relapse after a transplant, and then are you going to need another transplant? That really seems like a bridge too far for a lot of our patients.
Well, we're awaiting any other questions. I was going to ask.
Oh,
I'm sorry. Apologies.
I was just going to agree with what Dr. Grupp said. I think for patients that lose their B-cell aplasia or have short-term CAR-T cell persistence, that there does seem to be a role for transplant in abrogating that increased risk of relapse. And I agree that it's difficult to compare trial to trial. Also, these small number of subjects, and I think a larger study is needed. We did see a benefit of patients undergoing first transplant, and because we did continue to see long-term relapses within our subject population.
And to answer your question, I think it's important just to preface that not necessarily all CAR-T are the same. They are different drugs. There's different constructs. There's different activating areas. I mean, they do happen to see the same antigen, but I think they really have different biologic differences. So when we as clinicians we're all clinicians, we sit and choose, ultimately a choice of choosing a drug, we're going to look at a safety profile, an efficacy, and we're going to look at a toxicity and a cost. Those 3 things will drive decisions. So I think that, yes, there's no question in my mind that we're seeing CD19-driven responses in large cell lymphoma. I think the axicabtagene ciloleucel studies from Gilead Kite have a very similar long-term outcome. About the same, that doubling of the predicted natural history. Early on, if you look, they have 90% response rate. Well, there's a different selection criteria, there's a different trial design, and there's a different amount of the lymphodepleting chemotherapy that's going to affect early detection. It's long-term that we look at. I think the work coming out of Celgene Juno also has a very, very similar, in that 30 to 40% approximate long-term progression-free. They all look efficacious. They all are changing in natural history. I think what we're going to see, the future, the comment is, I think we're going to use toxicity and costs to actually start to segregate. And I also think that new studies and new explorations, as we just heard from Jordan and Shannon, will also affect, how we use these drugs, not just 5 years from now, but probably 12 to 18 months.
And I'm going to jump in and make a very quick comment. One of the things that drives people crazy listening to these sorts of discussions is that we all use different grading scales for different toxicities, and I think that fortunately is over. The ASBMT put together a huge group of investigators across all the different trials and all the different commercial products, and has come together with a consensus grading scheme that I think will be published very soon, and we're very hopeful that everybody, both commercially and in research trials, will be using the same grading scales for CRS and for neurotoxicity in the very near future, and that may help some. I'm sorry?
The new scale, is that CAR-Tox? What is that?
Right now it's being called the ASBMT toxicity grading scale, but it's not published yet. We're complete with the process. It has been presented publicly, and this should be coming out very soon. So.
It's a consensus management tool. Yes.
Please.
Hi, I'm Andy Jackson from Scrip and the Pink Sheet. Since you've brought up now multiple times the issue of cost, can some of the panelists please comment on what you're seeing in practice now, how cost and reimbursement is holding things up or not in treatment, and how you expect that to change once you start adding on combinations? Pembrolizumab also is not an inexpensive therapy.
So I think we can just go across the group here, because each perspective differs a little bit. So from pediatrics, there's only one approved product. Obviously we're hoping that we're going to be able to replace transplant with that approved product in a substantial fraction of patients, which I think is part of the value proposition. We don't have to worry about Medicare in pediatrics, so what we deal with is commercial insurance and Medicaid, and we're not having a lot of problems getting this paid for by commercial insurance at all in our experience. We gave the first commercial product ever, and we've prescribed probably more of it than most centers at this point. And so commercial insurance is not an issue. Medicaid, every single state has multiple Medicaid authorities, and you're dealing with every single one, and you're inventing the wheel every time. But the reality is that we're getting most of those paid for as well. And so from a reimbursement of the actual product, I think it's going okay. From a reimbursement of the hospital costs, since this is not DRG based, this doesn't seem to be a major problem in most instances, although there are always state Medicaids where there's a challenge. So in the end, our ability to treat these patients has not been compromised by insurance issues at this particular moment in time, and that's very pediatric specific.
So from the adult side, so I'll also say I am for the former ASBMT and now TCT, Transplant and Cell Therapy group. I sit as chair of the Value and Health Economics Interest Group, and this has been a big highlight. And yes, private insurances are covering this, and it's often a journal voucher through direct cost of the drug, and with a treatment with coverage for the care of the patient. The issue is what Steve mentioned is absolutely true is Medicare, because right now, and it's not that I don't think that it's reticent or it can't be fixed. It's just that the treatments are moving faster than our health system can evolve. The way Medicare reimburses, they reimburse a daily payment for outpatient care, and they reimburse a bundle for inpatient care. With a lot of lobbying, there was just a request to change to create a code for the coverage of CAR-T. It got added on to an autologous transplant. So for a drug that costs $373,000, both companies, for a large cell lymphoma, the rate previously was $8,000 to $18,000. Now it's up to $38,000 is what the hospital can expect to get from Medicare. So the ability to manage a patient who requires inpatient hospitalization at the time of application of the drug is daunting. It is doable, but it certainly will be more feasible if you have an agent that can be used effectively in the outpatient setting where you can be assured reimbursement. And that's when I mentioned earlier the question is, we're going to look at all of these drugs independently and evaluate toxicity profiles, costs, and our ability to actually use them in a patient.
So I was just going to add a little bit of perspective to that. I've spent some time with ASH on Capitol Hill and meeting with members of CMS, and the challenge is that, again, I believe the technology has gone faster than our language within CMS rules to deliver this. So there are payment systems that are challenging. For those institutions that are paid under the prospective payment system, there's a DRG of roughly $38,000 for that episode of care. And then those organizations may also receive a new technology add-on payment that covers a half or less of the $373,000. So for those institutions, there's a financial loss of $200,000 plus to care for an inpatient on a per-patient basis. For those institutions that are PPS exempt, the 11 cancer specialty hospitals, there's no new technology add-on payment, so there's no coverage for the cost of product procurement on the inpatient basis, and then each institution is covered under a separate, what's called a TEFRA cap. So it's not a sustainable reimbursement system, and I think you'll find many of us speaking with CMS and ASH and ASBMT, the Alliance of Dedicated Cancer Centers, and others have been collaborated trying to work with CMS to create a new language of reimbursement, because you may find when you look at this, and there was an abstract earlier this year, that the aggregate cost of taking care of people with third and fourth line therapy for diffuse large B-cell lymphoma probably exceeds that of delivering CAR-T cells. It's parted out in such a way that seems to better suit CMS's way of paying for services. I think the cost of the product acquisition coupled with the cost of care, managing toxicities like cytokine release or neurological toxicities, are something that has not been seen before, and for CMS this is uncharted territory.
Yeah, if I could just add one little more thing to that is, there's a way that with the hospital loss, you can actually, based on your charge ratio, cost to charge ratio, you can recapture some loss. And so what is being, you can see there are public statements that perhaps the cost of the drug should be right up, the charge for the drug should be $2 million. Now nobody's not going to, insurers you can negotiate and talk to, but if you can't pre-authorize, how can you have a debate? And so by doing this, you can actually, you can balance your losses to potentially try to decrease that, internal hit if someone has to be hospitalized.
But I would actually offer a different perspective on that. And again, I'm speaking Medicare as a pediatrician. And in the end, there's what hospitals charge for things and what it costs to do at the hospital differ very significantly and are nearly impossible to understand for somebody who went to grad school and really doesn't know anything about this stuff. But the cost of the product, that's actual money out the door. You have to pay for that. And so the approach in Medicare that has been most successful has been to just say, this is what it costs. We need this amount of money to cover it, and this does not apply to Medicare. I want to be very clear about that. But to try to top up or to go around Robin Hood's barn to cover this, I mean, in the end, the product costs what it costs. It is complete transparency on what the product costs, and that has to come back to the institution because they are not going to subsidize this.
I'd like to move in a slightly different direction with the question I want to ask the panel. We heard about MRD. In a number of these trials. That was very exciting to me. So how would you factor using MRD for trial design, particularly if you're looking at adjuncts to CAR-T cells, either nivolumab or pembrolizumab or even a transplant? Would you consider that in trial design?
I think it's something that's going to be interesting for us to study in future trials and see, is MRD going to be an early marker and maybe a surrogate of what we would see at 1 year for relapse-free survival. And if it is something that can correlate, then it may be useful in making decisions on who needs further therapy.
Wonderful.
And I would totally agree with that and say that right now the next-gen sequencing, which is probably where we're all going to be in 3 years or so, is something where we don't know what the clinical outcomes are. This is the first time anybody's looking at this, especially in the CAR-T cell front. We're not the only people who are doing this by any means. And so this is a chance to really understand how we can use this. But if we really are seeing a group that has an 80% duration of remission if they're NGS negative, then that may clarify the transplant discussion a little bit.
Another question there. So you're starting to hear technologies being used in combination. So you just heard about genomic testing, you're hearing about immunotherapeutics, and this is part of the theme of the meeting where you're beginning to see these things that had previously been siloed converge in terms of supporting what I'd say is very high complexity decision making, which is very exciting to see this all gelled together. Please.
Thank you. Jacob Please from Vantage in London. Just on that combination theme, a quick question for Dr. Gauthier, please. It's a very interesting data, but what surprises me is that JCAR-14 is actually not a CAR-T project that's being taken forward. In the Transcend CLL study, it's actually JCAR-17 that's going to be used. Why is that? Is the manufacturing process for JCAR-14 deemed to be too complex, or what's the reason?
So I don't want to talk too much on behalf of Juno, because I'm not on any advisory boards. I'm not. If I were, I would probably not be allowed to tell you. But so my feeling is that there's a significant change in the manufacturing, which is the LCL stimulation. So sorry for people that it's very technical term, which is basically you co-culture the CAR-T cells with a cell line that express CD19 and which helps the cells grow ex vivo. And I think, to me, that's the key difference between the two products. The transgene itself is the same, but within the manufacturing, there are probably other differences, maybe the use of different cytokines or maybe more subtle differences in the manufacturing, but the key difference is this LCL stimulation. And I think in the back of the mind, there's not a lot of solid data, but in the field, we had the feeling was that you would drive the T cells to a very differentiated effector state, which may be associated with toxicity. So that was, I think that was the hypothesis. And I think what we're seeing with Transcend in terms of toxicity seems very promising. So is it the only reason why? I don't know, but just highlighting this difference.
We'll have to take our last question here. Any takers? Yes, please.
Lynn Peterson with Trends in Medicine. So I'm going to come back to cost. Are you saying that with commercial payers, hospitals are actually coming out okay using CAR-T, but with everything else, they're losing money, or are they losing money on all these patients for now?
Yeah, and there's a difference, I think, in the adult experience and the pediatric experience. What I say, I base solely on the 30-odd patients that we've treated with commercial product since approval. And the answer is that we're getting paid for the product, regardless of the insurance. And I think our hospital has not been presented with a choice of subsidizing the product, but I think they would be reluctant to do that. They would see that as a money-losing proposition, but it hasn't come up. And while I don't speak for Novartis in any way, I do want to point out that in leukemia, if the patient doesn't receive insurance approval, the company will actually provide the product for free. And so it really, that issue just hasn't come back up in the pediatric setting, either with commercial insurance or with Medicaid. Medicare, I'll turn that over.
I think the private insurance in the adults, that from our experience, is not a cancer, we're not a cancer center exempt institution. We have not, it is a difficult undertaking to actually manage a patient. It can be done successfully as an outpatient. It can be done successfully as an inpatient, but I think there is financial risk in the care of Medicare patients currently. We all are waiting. ASH and ASBMT are both, bringing information to CMS. CMS has proven to be a willing partner to listen. It's just that how fast it will take to change the rules, it's certainly not easy today, and you certainly cannot assume that if you took care of every patient who came in the door, that you would be able to remain financially solvent on every case.
So I'll just add to that, on the commercial payer side, these are often things that are agreed upon with the payer as single patient agreements, so it's hard to speak categorically as to whether or not all of those are financially sustainable. In the case of CMS, and again, I work at a PPS-exempt cancer hospital, there's zero reimbursement for the acquisition of the CAR-T cell product, which means that before you've started treating the patient, you've already lost $373,000. And on the clinical care reimbursement, there's probably a per-patient loss for virtually every patient on inpatient side.
Pardon me? So, I'm sorry, I don't want to. No, you put yourself at risk. So there's a lot of interesting rules, and I'm not a Medicare expert, but I do know that what depends what an inpatient reimbursement is a bundle. And right now, this is a treatment for lymphoma. And so you have a potential bundle. I think if the patient gets admitted for a complication of a procedure, that's a different DRG. But what is the definition of an inpatient? It's 72 hours from anything that's within 72 hours. Sorry, the definition of an inpatient is 2 midnights. And anything that has happened 72 hours before those 2 midnights is incorporated within the cost of the bundle. So if you administer a product in the outpatient setting, on a daily basis, you may be able to get reimbursed. But if a person has a complication, and that's why we pay a lot of attention to complications, if they have a complication within 60 hours, you have to be prepared that the cost of the drug that you administer in the outpatient setting will be applied to the inpatient bundle.
It's very complicated, and I think none of us imagined that we'd be memorizing at any point in our lives 2 midnight rules or reading through 1,000-page long CMS documents and rules and proposals, but that's what life has come to at times. I thank you very much for your extraordinary questions. Again, I want to congratulate the presenters on some really amazing work on behalf of myself and Ash. You and your groups have done an extraordinary job in advancing the state of the art for patients and their families. We really appreciate what you've done to bring this to reality. Thank you again.
Thank you.
Hi. I'm with you. Oh, hi.